evening. I hope you guys are doing really well and enjoyed the first PowerPoint on biological molecules. And now we're on to the second PowerPoint of week 2. I know it's a hectic week, eh? Just the way it goes sometimes. I'm Ian Coates, and I'll be your host for Biology 1021, week 2, Biology of Infectious Diseases. All right, we're going to deal with fungi, bacteria, we're going to deal with viruses. I'll even talk about a prion. Lots of good stuff. Let's go. So, when we're talking about bacteria and viruses here, basically what we're talking about are microbes. And these are microscopic organisms. Okay, things there that we cannot see with our naked eye. We need a microscope there in order to see. Widely distributed in the environment, on inanimate objects and living things. They are everywhere. We are completely covered in microbes. All right. We will never ever get rid of these microbes. They were around billions of years before us. They'll be here a billion years after. The only way we are able to survive is to coexist, because 99% of them are good for us. They don't harm us. We utilize them there for yogurt, cheese, bread, beer, wine, pickled foods. One of my favorite foods is kimchi. Yum, yum, yum. Completely fermented. Fermented food is very good for you, actually. A lot of drugs are made by bacteria. Okay, we produce insulin. Okay, in big vats made by bacterium. Without the activity of decomposers such as bacteria and these guys here, the earth wouldn't survive at all. They provide too much food to too many creatures. Okay. Pathogen is any type of disease-causing agent. Bacteria, viruses, and others, they can be pathogens, or the majority of them are not. Defense against pathogens, we have the first, second, third line of defenses, which you'll get into there next semester in Bio 1022. But the first one there, barrier to prevent pathogens from entering the body. Skin, hair, mucous membranes. They don't care what microorganisms they're killing, they're just killing them. That's the first line of defense. There's phagocytic white blood cells, T cells. These ones here, they kind of resemble Pac-Man. Chomp, chomp. They digest these creatures, okay, that get into us. They break them down, kill them. And there is also the acquired and learned defenses. This is the immune system, where a particular disease sets off an immune response in your body. And if you survive that illness, you will never get the same illness again, because your body will remember it. Bacteria are single-celled prokaryotic organisms before the nucleus, you'll remember. 3 common shapes are cocci, sphere-shaped, bacilli, rod-shaped, and spirillum, curved or spiral. Okay. Most bacterium have a cell wall in addition to a plasma membrane. The cell walls contain a chemical called peptidoglycan, which is a disaccharide double sugar with an amino group. Some antibiotics, such as penicillin, interfere with the production of peptidoglycan. So that's why penicillin is an effective antibiotic, because it destroys the cell walls of bacteria, and then all of their contents spills out. Some are surrounded by a capsule that allows bacteria to stick to the surface and protects them. Bacteria are classified by differences in their cell walls, which are detected using Gram stain. Cell walls that have a thick layer of peptidoglycan stain purple with Gram stain, they are called Gram-positive bacteria. Gram-positive are purple. If there is no peptidoglycan layer within the cell wall, the cells stain pink and are considered gram-negative. Pink is gram-negative. These bacteria have an outer membrane with lipopolysaccharides. Lipo, fat, poly, many, saccharide, sugar, which are released when cells are killed by the immune system, stimulating inflammation and fever. Typical shapes of bacteria you'll see here. So what we've got going on here, this is what we call like a typical bacteria. You'll see there that it's got the flagellum there for motility. You have the fimbriae there that allow for, they tend to be sticky and allow to adhere to surfaces. There's your genetic material in the middle there, and you'll see there that it's just floating around. But then you'll also see there's another little bit of genetic material in here, and this is called the plasmid. This is the DNA or RNA, the genetic material, but then you'll see these round circular DNA bits. These ones right here, these are the kicker. Bacteria can share these. So if one bacteria is immune to a particular, antibiotic, all it takes is it makes a copy of this, sends it over to a buddy, and now that buddy bacteria is now immune. That's as quick as it takes. That's why these guys have been around for billions of years, because they're so primitive, they're so efficient. The life cycle of a bacteria, 20 minutes. It's crazy. 20 minutes and you now have an offspring. So you have a look here. There's A, B, C, and D. So this is a typical bacterium here. There's your Staphylococcus aureus down here. Round staph is round. Okay, Staphylococcus. There's your cocci, which is round. Here we've got rod-shaped bacilli bacteria. These ones here cause urinary tract infections there. And here's your curved shaped. Okay, just different shots here. I wouldn't ask you any of the names there and what they do. You'll get that there in your nursing or paramedic. Motile bacteria usually have long, very thin appendages called flagella. Okay, it's just involved in moving and locomotion. Some of them there will have flagella on just one side. You could have flagella on the other side as well, so double-sided. You could have flagella all over, so this thing can move in any direction. Some bacteria have fimbriae, these stiff fibers that help them stick to surfaces. There's a pili, sometimes called a sex pili, which is an elongated hollow appendage there used for transferring DNA between different bacteria. Okay, genes that give bacteria resistance to antibiotics can be passed through a pilus through conjugation. The whole idea there of sharing information between one bacteria and another is called conjugation. DNA is packaged in a chromosome in the center of the cell. Many bacteria also have small circular pieces of DNA called plasmid DNA. Okay, and these are genes that give bacteria their resistance. Those circular DNA, these ones here are the pain. These ones here gives bacteria their edge. They're unstoppable. We can control them by using soap, alcohol greater than 70%, washing our hands, keeping counters clean, doorknobs, ATM buttons, all these kinds of things, but we'll never ever get rid of them. Okay, so these plasmid DNA, these are the genes that give bacteria their resistance to antibiotics. Okay, abuse of antibiotics by doctors increases the number of resistant bacteria strains because we're just exposing more of them to the drug unnecessarily. Doctors giving out antibiotics for virus infections doesn't make any sense. Okay? This is how bacteria reproduce, through binary fission. Okay, the single circular chromosome attached to the plasma membrane is copied. And you'll see there that the DNA is circular, so it has a start and a finish. It's very easy, not like our DNA. Okay, it's a double helix. It's very difficult to copy it, replicate it. Then the chromosomes are separated as the cells enlarge, and the cell separates into two identical cells. Can reproduce rapidly under favorable conditions. Some can double their numbers every 20 minutes. Crazy, eh? So you go from 1 to 2, 2 to 4, 4 to 8. So within an hour, you can have one bacteria. So within an hour, you'll have 2, 4, 8. Yeah, they reproduce very quickly. Strep throat, tuberculosis, gangrene, gonorrhea, syphilis are well-known bacterial diseases. Not only does growth of bacteria cause disease, but some bacteria release toxins that inhibit cellular metabolism. A tetanus shot protects against Clostridium tetani, which produces a toxin that prevents relaxation of muscles. In time, all the muscles contract, causing suffocation. Devastating. Viruses, different ball game. If a bacteria, is this big here, a virus very much smaller. Okay, a virus would land on a bacteria. Okay, so bacteria are much bigger than viruses. Viruses very small, non-living, acellular. We don't even think that they're alive because we saw there last week our definitions of living. These creatures have to move, they have to grow, they have to reproduce, they have to absorb and digest. Viruses don't do any of these. Must reproduce inside of a host cell. They don't even have organelles in order to do anything. All a virus does is it lands on a cell and then injects its genetic material in, and that's it. 2 parts. There's an outer protein coat called a capsid and nucleic acid. It'll be RNA or DNA inside. That's it. Carry the genetic information needed to reproduce. In contrast to cellular organisms, viral genetic material can be DNA or RNA. And remember, RNA being the flimsy paper bag copy. But again, it works for them. Cellular parasites, all they do is they commandeer the metabolic machinery of a host cell. So once a cell gets infected, it's not your cell anymore. It's the viruses. And as you can see there from this slide here, just kind of in a comparative analysis between a eukaryotic cell. This is us, okay, with all of our organelles, mitochondria, okay, you got the cytoskeleton, microtubules, Golgi apparatus, there's your ribosomes there, rough ER, there's your nucleus. Here is a bacteria, much smaller, much more simpler, and here's a virus, even simpler on that scale as well. Okay, interesting. So just getting into the whole idea there, when you're talking a virus, there's your capsid. That's all it is, just an outer coat, outer shell, and your DNA in the middle. That's it. It just lands and injects its DNA into the host cell, and that's it. And it lives on. So despite all their diversity, all viruses have an outer capsid composed of a protein subunit and a nucleic acid core, composed of either DNA or RNA, but not both. Adenoviruses cause colds. Okay. Portions of the virus bind to the receptor on the host cell's surface. Once the virus attaches, its DNA or RNA enters the cell and codes for the protein in the capsid. The virus may have genes that code for enzymes needed for the virus to reproduce and exit the host cell. A virus relies on the host's enzymes and ribosomes for its own reproductive purposes. So, typical virus structure, again, there's your capsid, okay? In this case here, we're dealing with the influenza virus. This is RNA in here. It doesn't even have DNA, okay? It's just flimsy paper bag genetic material, gets injected into a host cell, and then the host cell's organelles begin manufacturing more virus. Okay. Bacteriophage. This is a virus. I know it has bacteria in it, but this is a virus that is only goes after bacteria. So this is a virus that only goes after bacteria, bacteriophage. Sometimes the virus kills the infected host in which it is replicating. This is called a lytic cycle, when the virus kills the infected host, which really doesn't do much, because you want that host to be your home. But sometimes the virus kills the bacteria. This is called the amount of time is called the lytic cycle. At other times, the virus integrates itself into the host genome but does not replicate. This is called the lysogenic cycle. Viruses that infect humans may have a lytic cycle or both lytic and lysogenic cycles. Lytic and lysogenic cycles, here we're talking about the attachment, where the protein on the virus capsid binds to a specific receptor. Then there is infiltration, where the host cell takes up the virus. Then there is replication. The virus uses the host cell organelles, nutrients, and ATP to make new virus particles. And then there is the assembly. New virus particles form and are released from the host cell, causing lysis and death of the host cell. And this is all demonstrated right in here in this diagram. If I could just blow it up a little bit here for your viewing pleasure, you'll see what's going on here, the whole idea. So you'll see there the whole idea, the virus attaching, injecting its genetic material. Okay, this is a bacteriophage because this is a bacteria. How do I know that this is a bacteria? Because the DNA is circular. Okay, and you'll see there, injects its genetic material. Okay, it can even be incorporated right into the bacterium. Okay, the bacterium DNA. And you'll see here, there's your lysogenic cycle. Moving into your lytic cycle here, you'll see there that this DNA can now be used, or the RNA or genetic material is now used by the cell to make more virus, and then cell death. There's your lytic cycle, where the cell dies. Here is where the genetic information has been incorporated right into the bacterium's genetic material. Okay, RNA viruses. These are viruses there that contain RNA instead of DNA. They must convert their RNA into DNA before it can be replicated within the host cell. RNA viruses mutate more rapidly because enzymes make mistakes during replication. They do not have any proofreading ability. Examples of RNA viruses: HIV, influenza, SARS, hepatitis, measles, polio, COVID, and Zika. Okay. The Zika virus, this is one there you want to take notice. Generally, it's been around the equatorial regions. It became a little bit well known there in Brazil. It's quite devastating there if this is contracted during pregnancy. Okay, and like an outbreak of the virus is affecting large parts of Latin America and the Caribbean, is likely to spread into the Americas. And I read a report there that there was reports of some Zika virus there within Florida. So anywhere equatorial, yeah, this is not one to get for sure. Okay, and it causes devastating effects there to the child. Okay, where the head isn't as at the proper size that it should be. Okay, and it's all spread through mosquito. Okay. HIV infects only certain white blood cells there within the human bloodstream. Okay, HIV is, this is, it's about as evil a disease as you can find. The only cells there designed to destroy HIV are the cells there that HIV targets. It's horrible. HIV infection leads to a low level of proper functioning immune cells. When the immune cell numbers decline below a critical level, the body becomes progressively more susceptible to what we call opportunistic infection. Most people who get HIV don't normally die from it. They'll die from the common cold or some strange form of cancer that nobody ever gets. Okay? If untreated, HIV eventually changes into AIDS, which is what we call acquired immunodeficiency syndrome. And here's the electron micrograph of HIV virus. Okay, budding there from a macrophage. Okay, you'll see right here, all these little guys in here. So structure of the HIV virus, okay, it's an RNA. Okay, so there's 2 strands of RNA and a capsid and these spike proteins. Okay, that's it. About as simple as you can get. Phases of an HIV infection. If the helper T cells are infected with HIV, the body can't produce antibodies and cytotoxic T cells can't destroy cells infected with the virus. This is what makes HIV so devastating. In the US, one of the most common causes of AIDS death, okay, is pneumocytosis pneumonia. Okay, in Africa, it's tuberculosis. See, pneumonia shouldn't be killing anybody, but to someone who has AIDS, they don't have an immune system. That's it, a simple pneumonia, it's lights out. Okay, not that that's a simple pneumonia, there's no such thing as a simple pneumonia, but you should be able to make it. But if you do not have an immune system due to AIDS, you're a sitting duck. Repeatedly hospitalized due to weight loss and constant fatigue, T-cell counts very low or has developed opportunistic infection from which they die, not from AIDS. Okay, so opportunistic infections include, there's your tuberculosis, Kaposi sarcoma, a cancer of the blood vessels, which tends to be very rare, invasive cervical cancer, yeah, everything else but AIDS. Global view of HIV infection. Okay, this is quite dated, but you'll see where the majority of them are. Okay, HIV initially was in the equatorial regions, but then it spread around the world. It is also a pandemic, just like COVID. Okay. Here's your COVID-19. Again, there's your RNA, your spike protein, your capsid, and your RNA in the middle. So just a nice little shot here of viruses. Here there's HIV, tissue infected, immune cells. Okay, and then there's a little right up there, HIV is an RNA virus that infects the CD4 immune cells. Okay, rhinoviruses, adenoviruses, respiratory. There's over 100 different viruses that cause the common cold, and that's why every time you get the common cold, you get a newer, updated version, because these RNA viruses, they mutate very quickly, which is why every winter we get a new cold virus. Okay. There's influenza, there's herpes zoster, which is also related there to shingles. Okay. Shingles there, also known as herpes zoster, is a viral disease characterized by a painful skin rash, okay, with blisters in a localized area. It is not herpes per se, but it comes from the same family of viruses. Okay. There's HPV. Okay, the whole idea of genital warts. We used to think that they were benign, but we started to realize that there was an increased association with cancer and HPV causes cancer. There are approximately 100 different strains of HPV virus that cause warts, with the majority causing no symptoms. Some strains cause warts, 30%, transmitted through sexual contact. This one here, you need to get vaccinated. Okay. Hepatitis. Okay, there's A, B, C, D, and E. You can get vaccinated for these ones too. I recommend doing that before you go down south and drinking contaminated water. Hepatitis C is a bad one for sure. Attacks the liver. There's rotaviruses, Epstein-Barr viruses. Please have a read of these for sure. It's all good for you. Infectious disease is classified as an epidemic if there are more cases than expected in a certain area for a certain period of time. So a few cases of a very rare disease may constitute an epidemic, whereas a large number of a very common disease may not. An outbreak, an epidemic confined to a local area. That is, the 2013 to 14 Ebola outbreak in West Africa was an outbreak. Global epidemics are called pandemics. HIV, AIDS, tuberculosis, malaria, influenza, COVID. Okay. Impacts of infectious disease, bubonic plague, which is still around today. I just read in the paper there a couple months ago. So this is August 2024. A couple months ago, maybe May 2024, there was some people went camping in Colorado and they contracted the Black Plague. It's not gone. It's still here. Okay, caused by the bacterium Yersinia pestis. Okay, and this wasn't, apparently the rats got a bad deal. It wasn't even the rats, it was the fleas on the rats that were carrying this bug. All right, so several pandemics of plagues have occurred throughout history. Estimated 50 million deaths. Nearly all of about half of Europe perished. Okay, and yep, not these guys here. It was the fleas on them carrying those bugs. All right. And you'll see there that the epidemics and how disease spread back in the day, quite different than how it goes on there today due to air travel. So if somebody has a disease in Australia, within 12 hours it'll be in Asia, and 24 hours it should be on the continental United States or North America. So, but you'll see there the hundreds of years, or the decades, I apologize there, the decades of moving, the waves of disease and how they moved, because, there wasn't any type of automated transport back then. Smallpox, caused by 2 airborne virus variants. Okay, deadly disease that in survivors can cause disfigurement and blindness. 500 million deaths worldwide in the 20th century. Eradicated in around 1980 through vaccination. But vaccination has to be maintained. One of the things there that came out of the pandemic was New York was measuring daily the amount of COVID in the sewage water. And guess what they found out one day? That there was polio in the sewage water in New York. So that means that polio, which was eradicated, 70 years ago, 60 years ago, is still around. Okay. And here's the eradication of smallpox, and you'll see there that there tends to be a line. You'll see here where the rich countries are okay, the lesser countries, with less cash available, they're the ones that tend to suffer. It's shocking, and you'll see this kind of a map many times over again when you're talking global diseases. It's a sad, sad state of affair. Ebola outbreak there in West Africa, as we said, 2013 to 14. This is an outbreak. Additional cases were reported in Nigeria and Senegal, with very isolated cases occurring in Spain, United States, UK, Mali, and Italy. TB used to be called consumption because it seemed to consume the patient from the inside until they wasted away. 1/3 of the world's population has been exposed to TB. This one here is a bad one. It's spread by close contact, droplets introduced into the air when an infected person coughs sneezes, okay, can float in the air for hours. Incubation period can be 4 to 12 weeks. Develops very slowly. The whole idea there that if you go and get a chest x-ray, x-rays only detect hard tissue. So a healthy set of lungs under x-ray, you shouldn't see anything. But lungs that have been exposed to tuberculosis actually show up on an x-ray because the tuberculosis creates these hardening nodules within the lungs themselves. Okay. Other white blood cells rush to the infected area together. They wall off the original infection site, producing small, hard nodules or tubercles in the lungs that are detectable by X-ray. The causative agent of tuberculosis, Mycobacterium tuberculosis here, shown here with a sputum sample. Sputum being the spitting up, the mucousy spitting up fluid from the lung. The whole idea there, whatever you spit out. And if you have a bacterial infection in the lung, it will be green color. Okay, you know what I'm talking about? That's what we call sputum. And there's your x-ray there. And you'll see these nodules here. That's from tuberculosis. Scarring. It's horrible what it does to the lungs. Symptoms, treatment of tuberculosis can take anywhere from 12 to 24 months of antibiotics. Okay? It takes a long time to get rid of this once it's been contracted. The tubercle liquefy and form a cavity. The bacteria can then spread from these cavities through the body and generally can be very fatal. A bad cough, chest pain, coughing up blood or sputum. As the disease progresses, the symptoms include fatigue, loss of appetite, chills, fever, and night sweats. Hence its original name of consumption. It just wastes you away. Okay. Malaria. A disease vector is a living organism that transfers the pathogen from one host to another. In this case here, for malaria, the vector, mosquito. The parasites that cause malaria belong to the genus Plasmodium, which are protists and infect the liver cells and red blood cells. Symptoms range from very mild to fatal. Most develop a flu-like illness, chills, fever, sweating, exhibit a cyclic pattern every 24 to 72 hours. Mosquito nets and anti-malarial drugs are used to prevent and treat malaria. And a nice little shot here detailing of how the malaria bug gets into us. Okay, you'll see there a female bites and sucks blood. Okay, the Plasmodium. Okay, and the whole idea of a mosquito bites you and sticks its proboscis in you, the itchy, that's the saliva, and it has an anticoagulant, so the blood doesn't start to coagulate, so that the mosquito can continually suck up blood. And in this process right here, it transfers this microbe into us. And when the infected mosquito bites a human, it injects the bug into us, into the blood, which carries it there to the liver, okay, where they produce asexually in the liver, each producing many of its own kind. Yeah, this is a bad one as well, okay? And you see there's the process, all right? Have a look at that. And again, do you see the same line? Worldwide malaria cases, okay? Now, generally this could be for climatic as well, okay? But you'll see where the malaria is, okay? Influenza affects 5 to 20% of Americans each year and causes an estimated 36,000 fatalities. Okay, viral infections that cause runny nose, cough, chills, fever, head and body aches, nausea. You can catch it by inhaling a virus-laden droplet. Okay. Emerging diseases are diseases that occur for the first time in humans, are rapidly becoming more common in humans, or are entering into new geographical regions. That is avian flu, H5N1, swine flu, H1N1, Middle East, respiratory syndrome, MERS, Helicobacter pylori. Remember that one there? That one there is involved in stomach ulcers, surprisingly. So MERS is a type of coronavirus which are known to cause respiratory problems. Precise animal host is unknown but has been found in camels and bats. Can be transmitted between individuals by close contact, causes pneumonia and kidney failure, and has a 30% mortality rate, which is very significant. Deadly. Antibiotic resistance. This is a huge problem. And you see, look at this. And just jumping here, here we have normal bugs, and we have two bugs here, bacteria, I call them bugs, bacteria, 3 syllables, bug 1. So you have two bugs here that are antibiotic resistant, okay? But they're surrounded by all these guys here, so these guys here should be able to outmuscle these two, okay? And that's generally how the world works. But what happens there when the doctor prescribes antibiotics needlessly, you're killing all the good bugs in your colon. You see, you take the antibiotic, and what it does is it wipes out all the good bugs and it leaves these ones here behind. So this is the trouble. So if you have a virus and the doctor gives you an antibiotic, you ask the doctor, why are you giving me antibiotics? Antibiotics only kill bacteria. I have a virus. Don't. We don't want to needlessly expose more microbes to these antibiotics than needed. Right? Makes good sense. Tuberculosis, malaria, gonorrhea, Staphylococcus aureus, and enterococci. There are a few of the organisms that have developed antibiotic resistance. Okay, more and more organisms are becoming multi-drug resistant. There's C. difficile. There's MRSA. Like if you get these ones here, there's really not much you can do. Okay. 4 more infected by superbugs. You're hearing about this more and more. Okay, so you guys know now, if you have a viral infection, nothing stops that other than good sleep, generally speaking. Bacterial infections, antibiotics, but use them sparingly. Just more on antibiotic resistance. There's your MRSA. Okay, there are different grades of antibiotics, the lowest level being penicillin. The highest level, vancomycin. This is the strongest antibiotic known to humankind. And when I was your age, this killed everything. Does it kill everything now? No, it doesn't, because some bacteria like MRSA or vancomycin-resistant enterococcus, they don't respond. They do not respond to vancomycin treatment anymore. What do you do? What do you do if you get MRSA? You pray. Prevention of antibiotic resistance. Take all the antibiotics prescribed. You need to kill all the bacteria. That's why if a doctor prescribes 10 days of antibiotics, you take them for 10. Because you got to get rid of everything that's supposed to be gone. You don't want this thing coming back. You don't want any of these antibiotic resistant bugs to somehow squeak by. Do not take antibiotics for viral infections. Do not save unused antibiotics or take antibiotics prescribed for a different infection. Each year in the U.S., 2 million people become infected with antibiotic-resistant bacteria, resulting in 23,000 deaths. Okay? And there's a nice little summary on bacteria. 99% of bacteria are good for you. Involved in the normal body flora. They are good for us. There's more a little summary on viruses, fungi, parasites, and the questions involved. Have some fun with these, everybody. Pause and start the video as you need. Go back and hit some of these questions. They're all lots of fun. I hope you had a great week too. If you're just watching this video here, please remember there's another video there on biological molecules involved in the week 2 PowerPoint system as well. So this week there's 2 PowerPoints. Make sure you've watched both videos and read it all. Thank you so much, and we'll see you for week 3. Talk to you later. Bye-bye.